Hello viewers, and welcome. Thank you for your interest in this presentation. I'm Adam Zimmerman, and I'm a master's candidate in the Biosystems Engineering and Soil Science Department in Knoxville. Dr. Forbes Walker is my primary advisor. Today, I will be presenting about my ongoing thesis research project. Going forward, I will discontinue the use of the video of me so that it does not take away from the presentation. Now, let's get started. We all know that climate change is an intensely debated topic in modern times, and no matter which side of the argument you find yourself on, I hope we agree that finding solutions to these issues is an important venture. My goal is to be able to help producers reduce ponding in fields or subpar yields from drought-stressed crops. To date, the focus of my research has been to see if we can identify a method that will preempt the effects of flooding that some producers must deal with by infiltrating that water into the soil profile, in hopes that what we store in the soil will be accessible to crops later in the growing season when drought stress can negatively impact crop yields. Before I get too far into things, let's lay some groundwork for understanding. Infiltration can have many different meanings depending on the context, but for this project, we will focus on saturated hydraulic conductivity, which is defined as the ability of a soil to transmit water under a gradient. This concept is based on Darcy's law, which details how fluids can move through a porous medium. Don't worry, I won't bore you with the complex equations or time-consuming calculations. Just know that saturated hydraulic conductivity is very much determined by the texture of the soil. This is because saturated hydraulic conductivity, which is often denoted as KSAT, KFS, or KS, is a value which corresponds to how much water a given soil can move when that profile is completely saturated or at field capacity for soil moisture. I'd like to draw your attention to the table I made based on an online USDA NRCS chart, which gives estimated infiltration values based on texture. Just note the differences between texture and how the infiltration rate, estimated of course, can vary. Now more groundwork. The instrument I have been using to obtain my data is a Saturo dual head infiltrometer by Meter Group out of Pullman, Washington. It functions as an automated single ring infiltrometer with two different pressure head levels. One nice thing about these instruments is the reduction of human error in the sampling process compared with other infiltration measurement methods. This device uses a computerized control system to incrementally add water as needed to maintain a constant hydraulic head. It also charts the water level, pressure head, and instantaneous flux, and provides all of this in a minute-by-minute output on an Excel spreadsheet, along with the final KSAT value. The chart on the right is the only input I have over the device, other than insertion of the ring, which is very simple. As you can see, this process is quite time-consuming when you recognize that the runtime of each observation is between 75 and 180 minutes. Another nice feature of these devices is the ability to function without supervision, which allows users to accomplish other tasks, thereby increasing efficiency of labor and time. Now let's continue to lay more groundwork. Cover crops are typically defined as non-economic crop grown between harvest and planting of cash crops. There are many different species and varieties of cover crops to choose from. Cover crops have many benefits, or co-benefits also, such as reduction of soil erosion, trapping of excess nutrients, fixing nitrogen for legumes, adding organic matter to the soil, even reducing compaction and mitigation of plow pans. There are also many ecological or ecosystem services provided by cover crops, which can benefit wildlife and pollinators of the surrounding area. Sometimes, producers can even harvest some cover crops, such as winter wheat, which can add to the farm revenue for the year. I will mention that some work is currently being done to look at the forage benefits of certain cover crops in hopes that some producers may find another value-added benefit of these cover crops by grazing cattle to remove residue and in turn have some fertilization through manure. Your local extension and NRCS offices should have resources available to most producers to assist with cover crop selection and/or planting, should you so be interested. So how would cover crops help to increase infiltration? Well, they would function much the same way as what is depicted on the graphic. The roots they grow will become pathways for rainfall to flow into the soil profile once they decompose. And as mentioned previously, they can also fight compaction, especially using brassica cover crops like tillage radishes. The increase in organic matter from the cover crop residue can lead to increased aggregation in the soil profile, which can increase the water holding capacity of the soil as well. The microbes and other small soil critters are happier, and generally speaking, the overall health of the soil is better and more resilient. These processes are also a function of no-till systems because the elimination of tillage allows these beneficial processes to take place. This is important because as earthworms and other small soil critters transform soil profiles, the soil can move much more water through preferential flow paths. If I'm remembering correctly, Pazul's law for laminar flow states that a doubling of a channel's radius equates to a 16 times increase in flow potential. Simplified, that means if a little worm pathway that transmits water has a radius of 1 millimeter and is expanded to 2 millimeters by a bigger earthworm or other soil bug, that single channel can now transmit 16 times more water than before. Just imagine all of this happening at field scale. To simplify this slide, we use some research plots at MTREC in Spring Hill and at UTREC-M in Milan, which have various cover crop treatments. First, we sampled the plots with the Saturo infiltration instrument to determine if any of the cover crop treatments show significant infiltration increases compared to the control. If more than one treatment did increase infiltration, we would then compare the treatments against each other to see which one did a better job. And third, we would compare the two locations, Spring Hill and Milan, against each other to attempt to evaluate any significant differences exhibited by the difference in time scale between the locations. And finally, we would collect yield data from the cash crops grown on these plots to evaluate if any of the cover crops were having yield impacts due to the cover crop treatment. As mentioned, our research is being conducted at two locations, MTREC in Spring Hill and UTREC-M in Milan. Both locations are managed as no-till corn-soybean rotations grown under natural precipitation situations. All plots are approximately 10 feet by 30 feet. When it comes to cash crops, they are managed like any typical producer would manage a field of row crops in a soy-corn rotation. Here's the plot map for MTREC. This is the second year that these plots have been managed. And as you can see, the treatments are a combination of single species, double species, and an NRCS recommended soil health mix containing 6 species. At this location, we are sampling 2 soil health mix treatments because the main ingredient varies between cereal rye and winter wheat, which are 2 of the most common grass species used for cover crops. Many producers are using crimson clover to fix nitrogen prior to planting corn in hopes of reducing nitrogen input costs. This is why we chose the winter wheat crimson clover treatment to sample. Here are a few pictures from our research plots at MTREC in Spring Hill. These plots are under their second year of management in a corn-soybean rotation. They were cropped with soybeans last year, so they are planted in corn this season. These photos were taken in early March, just before we finished collecting data at this location for the season. Note the large variance of ground cover for all of the different species and varieties given in these blocks. Here's the plot map for UTREC-M. This is the 7th year that these plots have been managed. These plots are pretty comparable to the MTREC plots, considering that they are also comprised of single species, double species, and an NRCS soil health mix that contains 5 species. The treatments are just slightly different, but function essentially the same. When these plots were installed, they were also to incorporate a planting method study, hence the DB, which stands for drilled versus broadcast methodologies. While I recognize the small variations in cover crop species, I believe that these treatments are still comparable to the MTREC plots because of the similar functionality of the cover crop treatment species. And here are a few pictures of our research plots at UTREC-M in Milan. These plots are under their 7th year of management in a corn-soybean rotation. They were cropped with corn last year, so they are planted in soybeans this season. And these photos were taken in May of 2020, just prior to burn down before planting. Look at the large variation in growth and species of all these different cover crops. Unfortunately, the preliminary analysis does not show that there were any statistically significant differences among cover crop treatments for our research plots when tested at a 95% confidence level. So instead of results graphs, I wanted to share with you the variability of the data that we have been experiencing. For the sake of viewability, the details on the screen, given our current format, I have combined the median values from each plot and calculated averages of those medians based on cover crop treatment. I then color-coded them by location. The blue bars are from Spring Hill, the brown bars are from Milan. These results were quite surprising and prompted us to do a bit more research as to what could cause a lack of significance among the infiltration experiment. We discovered that the area in Milan has been no-till for over a decade, and that the area at Spring Hill was in a long-term sod trial before being converted to row crops almost 3 years ago. The significance of this became quite apparent after checking some scientific literature. And here's what we came up with. Based on our knowledge of the previous landscape use for the experiment locations, we now believe that we understand the underlying soil structure allowed earthworms to create an area where infiltration is dominated by preferential flow paths. I pulled this table from a master's degree thesis written in 1996 by plant science student Nathan J. Stork, who worked under the late and legendary Dr. Don Tyler. Basically, he concluded that between 1 year and 5 years after stopping tillage, that a significant difference in the quantity of earthworms takes place, but that result becomes less significant between 5 years and 10 years. He also noted large differences of earthworm counts between tilled and no-tilled ground. He also looked at some cover crops, but I believe concluded the differences to not be statistically significant. To summarize his work, based on my understanding, after a given time frame of stopping tillage, the earthworms tend to dominate the soil profiles and cause channels of preferential flow, like previously mentioned, which is why we believe we were not statistically significant differences among our cover crop treatments or plot locations or across the timescale incorporated in our infiltration research experiment. So now what? Now what will we focus on next? This demonstration was also done in an Ohio alfalfa field where they connected a pump to a field drain tile and added smoke to the airflow. It's expected that smoke would follow the drain tile system, but what wasn't expected was smoke to show up hundreds of feet away where there were no drain tiles. It was later determined that earthworms were the cause of the interconnected network of flow paths under this long-term perennial forage field. So, as of now, our future direction appears to involve using a new soil health field test, which is cheap, easy, and portable. but is also showing positive results in some long-term experiments being run by other researchers. So hopefully we can get going soon and collect more data for analysis. Here's our funding slide. We note no conflicts of interest. This concludes my presentation, and I'm so grateful and humbled by the opportunity. Again, thanks so much for your interest in this project and your attention during the video. Please send any questions or comments via email to myself or Dr. Walker. Our contact info is on the title slide. I hope everyone stays well and safe. Take care.